In a land far, far away, there were a group of monsters. Miss Monster has 4 children, but is a bit confused because none of them look like her. She decided to put her genetics knowledge to the test and evaluate a section of her own DNA and each of her children's DNA. She dug out her old genetics notebook and turned to a page that had all the information she needed in order to complete her genetic code sequencing. On the page, it says that a transition is due to a switch from either a purine to a purine or a pyrimidine to a pyrimidine. A transversion is due to a switch from purine to pyrimidine or vice versa. It also laid out the start and stop codons. The one start codon is AUG, and the 3 stop codons are UAA, UAG, and UGA. Lastly, it stated that the 4 kinds of point mutations. A missense mutation results in a change of an amino acid. A nonsense mutation changes the codon into a stop codon. A silent mutation results in coding for the same amino acid, and a read-through mutation changes the stop codon into an amino acid. Miss Monster decided to look at her own genetic code before tackling each of her 4 children's genetics codes. Her first step in the process was to find the start codon for a protein that coded for 4 amino acids. Remember, T's transcribe into U's when switching from DNA to RNA. She looked at her top strand of DNA from the left to right and found an ATG, but this isn't the one she was looking for, because the stop codon is more than 4 proteins away. She then looked at the bottom strand of her DNA and found another ATG, but this one isn't the one she's looking for either. Next, she looked at each DNA strand from right to left. Miss Monster found the correct ATG she was looking for on the bottom strand of DNA going right to left, meaning this will be the template strand and the top strand of DNA will be her coding strand. The next step is to transcribe her DNA sequence into mRNA. Miss Monster rewrote her DNA the 5' to 3' direction and changed all her T's into U's. Lastly, Miss Monster needed to translate the sequence to see what proteins it coded for. She found that her sequence coded for methionine, valine, serine, lysine, and then a stop codon. With this information, Miss Monster looked at her first child's matching DNA sequence. She noticed that there was a change from a thymine to a cytosine in the second codon, resulting in a change in the proteins coded. Miss Monster had a valine, and her first child has an alanine. This occurrence can be identified as a transition and a missense mutation since the coded protein changed into a different protein. Next, Miss Monster looked at her second child's DNA sequence. She noticed that in this one there was a change from adenine to thymine in the 4th codon. This change caused a lysine protein to change into a stop codon. This occurrence can be identified as a transversion and a nonsense mutation because the amino acid changed into a premature stop codon, thus altering the DNA. Next, Miss Monster looked at her third child's DNA sequence. She noticed that in this sequence there was a change from a cytosine to an adenine in the 2nd codon. Though there was a point mutation that occurred, the protein that was coded for did not change. This occurrence can be identified as a transversion and a silent mutation because the protein did not change. Lastly, Miss Monster looked at her 4th child's DNA sequence. She noticed that there was a change from a thymine to a guanine in the last codon. This change caused a stop codon to switch to a glutamic acid. This can be identified as a transversion and a read-through mutation because the stop codon changed into a coding amino acid, resulting in a longer protein sequence.